My name is Macharia Kang'iri, and I will present the study about a mathematical model predicting gliding speed of actin molecular shuttles over Myosin motors in the presence of defective motors. In the cell, materials needs to be moved and forces need to be generated. The figure on the left shows the cytoskeleton of a cell, fluorescently labeled and magnified under a microscope. Around the center, we see the nucleus of the cell. The green line shows the structure of the cytoskeleton, which is composed of various filaments. The figure on the right shows the motor proteins and filaments that facilitate movement, force generation, and the structure of the cell. The microtubule filament is around 25 nanometers. Kinesin and dynein transport materials by walking on a microtubule but towards the opposite direction to each other. Top right, we have actin filament, which is around 7 nanometers. Myosin slides on the actin filament to produce contraction force. We also have F1 ATPase, which produces circular movement. Despite being nanoscale in size, these systems are very efficient and they convert chemical energy directly to mechanical energy. No heating is involved and only adenosine triphosphate is consumed. In this study, we shall focus on myosin and actin due to its high speed. To take advantage of the attractive features of biomolecular motors, they can be extracted from the body and used on a substrate in a process called in vitro motility assay. This process is as shown here in the figure on the left. The movie on the right shows the in vitro motility assay viewed under the microscope. The moving white lines are the actin filaments being propelled by myosin motors. This in vitro motility assay setup can be utilized in various applications. Some of these applications include making biosensors and making biocomputers. For the biosensor application, shown here on the left, the basic working principle is that cargo or analyte is loaded on the filament. Then the cargo carrying filaments are fluorescently labeled to aid in movement tracing. The cargo or the analyte is then allowed to move, and when it reaches the detection point, the observed reflected light can indicate whether the cargo has been delivered or not. For the case of biocomputers, filaments translated by biomolecular motors and guided by microfabricated microchannels can be used for parallel computing, something that conventional silicon-based computers struggle to solve. The figure at the bottom center shows a typical concentrator device implemented by Macharia and others. The problem is that when myosin motors are adhering onto the surface of the substrate, they become denatured due to mutation or unfavorable binding on the surface of the substrate. I shall call such denatured myosins as a defective myosin or a defective motor, and a normal working myosin as an active myosin or an active motor. The defective myosin has been shown not to contribute to the progressive movement of the actin filament, as shown by the figure on the left, and sometimes impede the progressive movement of the actin filament. In our study, we developed a mathematical model and represented the defective myosin as a spring. As shown by the schematic on the right, when the defective myosin binds to the actin filament, it does not release the filament until it is forced by a greater force to detach. The reason why we used a mathematical model is so that we can focus on a few parameters and follow our study systematically, which would be difficult to do in an experiment. In addition, we can provide important insights before the actual experiment is conducted, and we can do so at high resolution. In the following slides, I will show how we modeled the defective motor behavior, how we evaluated the critical motor ratio, and how we checked for the stability of our system. For the force velocity relationship of our actin myosin system, the binding active myosin facilitates the forward movement of the actin filament. At the same time, the binding defective myosin impede the movement. If the impedance forces build up and reach to F stall, they will cause the actin filament to stop. This behavior is modeled by this equation here on the right. Looking at the figure on the left, we see that after the time tau 1, the defective motor binds to the actin filament, and this marks the beginning of time tau 2. The defective myosin stays attached to the actin filament, elongates for time tau 2 until the force reaches the rupture force, which forces it to detach. In the animation on the right, the blue spring represents the defective myosin and the green one represents the active myosin. The higher the speed of the actin filament, the shorter the time tau 2 is going to be. For simplicity, spontaneous dissociation of defective myosin from the actin filament was neglected. The binding rate is expressed as 1 over tau 1, and the duration that the defective myosin binds to the actin filament, which is tau 2, depends on the speed of the actin filament, the rupture force, and the spring constant. We then took the time average of friction forces generated by defective myosins as governed by this equation. Since the active and defective myosins are binding to the actin filament of the same length, the length parameter cancels out and the impedance per active myosin is given by this equation. The parameters used in our mathematical model are as shown in this table, and these parameters are taken from previous studies as shown here. We solved for these equations. The force velocity equation, the time averaged of the defective forces, considering the impedance per active myosin, and the product of the defective motor binding to the actin filament and the time averaged defective forces is equal to the product of the active binding motors and the force that facilitates the movement of the actin filament. The solutions to these equations are shown here by this plot. We solved for active ratio 0.8, 0.85, 0.9, and 0.95, and we overlaid with force velocity plot. The solutions can be summarized by 3 cases, as I will discuss next. The first case is above critical value, and there is no solution. The second case is at a critical value of the motor ratio, and there is only one solution. The third case has 2 solutions: the high-speed solution and the low-speed solution. We need to evaluate for the stable solution. At each speed, due to force fluctuations experienced by the actin filament, sometimes the speed can go up or can go down. At this high speed, if the speed goes up, the impedance increases, causing the filament to decelerate and go back to the initial velocity. If the speed goes down, the impedance decreases, causing the filament to accelerate and still go back to the initial velocity. This is a stable solution. At this low speed, if the actin speed goes up, the impedance decreases, causing the actin speed to increase more and more and does not go back to the initial speed. This solution is unstable. Taking the stable solutions and rupture force as 9.2 picon and motor ratio as binding active motors over the total binding motors, we find that at the critical motor ratio of 0.85, the actin speed increases abruptly. We solved for other rupture forces ranging from 2.0 to 8.0 picon. The lower the rupture force, the lower the demand for AC active ratio. In all cases, we found that when the respective critical motor ratio is reached, the speed of the actin filament increases abruptly. Our one-dimensional mathematical model is simplified and considered few factors. We are currently running a 3-dimensional simulation which takes into consideration the thermal fluctuations and the hydrolysis cycle of the Myosin motors. The results are similar in the sense that at low active motor ratio there is no movement, and at a critical motor ratio the actin filament velocity drastically increases. This is as summarized by the following movie. The red line represents the actin filament. The green dots represent the active myosins, and the few blue dots represents the defective myosin. At active ratio of 0.7, the actin filament stays stationary and does not move. At the active ratio of 0.92, the actin filament experiences jerky movement, and at the active ratio of 0.98, the actin filament experiences smooth gliding. There are insights about the actin filament transition mechanism and biosensor design that we can pick up from this study. First, the active motor ratio of the motility assay is a single important factor that influences the gliding onset of the actin filament, and in our case, we found that critical active motor ratio to be at around 80%. And second, the substrate material should be selected to minimize the denature of the Myosin motors, and that they should retain the activity of the available Myosin motors. Thank you.